Hello everyone. So I am here again. Today I will be talking about Helicos sequencing technology. But before I talk about Helicos sequencing technology, बेटा मुझे एक शिकायत है और वो शिकायत ये है कि you are 41 students in the class and out of those 45 students only 7 to 8 students are responding on the classroom dashboard. ओके तो गूगल क्लासरूम डैशबोर्ड में कुछ तो लिखिए, कुछ प्रॉब्लम्स हैं, कुछ क्वेश्चंस हैं, कुछ क्वेरीज हैं। आई एम गोइंग टू रिस्पॉन्ड अबाउट इट। आपको वीडियो अच्छा लगा वो लिखिए, बेकार लगा वो लिख लीजिए, ठीक है। सो एटलीस्ट अटेंडेंस ही लगा दीजिए कि यस मैम आई हैव गॉट इट एंड आई हैव सीन इट। एटलीस्ट आई गेट टू नो कि कितने बच्चों ने देख लिया है, किसको मटेरियल मिल गया है। If I am making an effort from my side, then I expect something from your side too. So kindly do that. If you do not do that, मैंने नेहा को भी बोल दिया, मैं वीडियो बनाने बंद कर दूंगी, नहीं भेजूंगी. Okay. Okay. Now moving forward with the Helicos sequencing. See, Helicos sequencing there, it is a sequencing technique which was started in 2003. April 2003. How does it came up? It came up as a publication which was in which Dr. Steve Quack was one of the co-authors. And after coming forward with this technology in 2003, December 2003, they founded a company called as the Helicos. Okay? This Helicos company became functional in 2004. However, this sequencing technology had certain limitations. The limitation was that each run was very expensive. Okay, therefore the technology overall was very expensive for the user, and it took a lot of time to deliver the data. That is why there were only few takers of this technology at that time. So they suffered quite a loss, and as a result, in 2012, the company filed bankruptcy. However, in 2013, a company called as Sequel, it was impressed by the technology that was used by Helicos, and they started to revive this technology again. So now they are in the process of doing so. Now coming to what is actually Helicos sequencing. Helicos sequencing belongs to the category of third generation sequencing. Why does it belong to the category of third generation sequencing? Is because this technology, it involves sequencing of a single molecule. Okay, that is important. So all the third generation sequencing, they use single molecule sequencing. Whereas how is it linked to the second generation sequencing is that like that of second generation sequencing, this technology also involves sequencing by synthesis, which means that nucleotides are incorporated to the templates complementary to the template strand. Okay, and there is synthesis of the new strands of DNA. Okay, now what is the difference between the Helicos sequencing and the second generation sequencing? The difference is, आप थोड़ी देर के लिए Illumina याद कीजिए. When you used to sequence on the Illumina platform, so all the 4 nucleotides ACTG, they were added at the same time during the sequencing reaction. But in this case of Helicos, only one type of nucleotide is added during the sequencing reaction. एक टाइम पे एक ही न्यूक्लियोटाइड जाएगा, ठीक है, दूसरा नहीं जाएगा. सो ये दो यूनिक कैरेक्टर्स हैं इसके. पहला the single molecule sequencing, and the second one is that a single nucleotide of nucleotide is added and during the one cycle. And what is the similarity? The similarity is that like that of Illumina sequencing, it is sequencing by synthesis, and another is that like that of Illumina, the nucleotides are fluorophore labeled. Okay, so these are the important comparisons between the two technologies. Moving forward, another characteristic feature of the Helicos sequencing, तो ये जो sequencing feature है बेटा, ये characteristic है Helicos का, और किसी और में नहीं मिलता है, and that is in addition to being fluorescently labeled, जो nucleotide use होते हैं during the sequencing process, they are called as virtual terminator nucleotide. अब ये है क्या? ठीक है, वर्चुअल टर्मिनेटर न्यूक्लियोटाइड का नाम बता रहे हैं. कि ये क्या है, एक ऐसा न्यूक्लियोटाइड है जो पॉलीमराइजेशन का टर्मिनेशन करेगा, मतलब ये ऐड हो जाएगा, इट विल बी इनकॉर्पोरेटेड इनटू द डीएनए एंड वंस इट इज इनकॉर्पोरेटेड इनटू द डीएनए इट विल नॉट लेट the sequencing go further. And how is that achieved? It is achieved by adding four specific types of fluorescently labeled nucleotides which are modified. These are called as modified analogs of the nucleotides. Now, what does it have? ठीक है, the each virtual terminator it has a free OH group उसमें होता है, ठीक है, जैसे नॉर्मल न्यूक्लियोटाइड में होता है which helps in the formation of the phosphodiester bond with the backbone of the DNA. Okay, however, it has a modification. The modification is that it has a linker which is connected to the nucleotide. ठीक है, सो न्यूक्लियोटाइड है N, इस न्यूक्लियोटाइड के साथ आपका एक ये लेट अस से दिस इज अ लिंकर मॉलिक्यूल and to this linker molecule is attached the fluorescent molecule along with the inhibitor. Okay, so we have a fluorescent ditethered to an inhibitor. तो fluorescence dye है which is attached to the inhibitor by the cleavable linker. Cleavable linker का मतलब है कि we can always cut the linker and let it go. So fluorescent molecule linker के साथ जुड़ा है एक inhibitor से. Okay, tethered to an inhibitor with the help of the linker. So these are the characteristic features and these are the modifications. ये आपको ध्यान रखनी है, but these modifications they help in the Helicos sequencing process. Okay, इस तरह के वर्चुअल टर्मिनेटर न्यूक्लियोटाइड का क्या एडवांटेज है? द एडवांटेज इज दैट कि 454 में अगर आपको याद होगा वन ऑफ द ग्रेटेस्ट लिमिटेशन ऑफ 454 वाज दैट कि जहां पर भी होमोपॉलीमर्स आते थे, मतलब बहुत सारे टी, बहुत सारे ए, बहुत सारे जे, सी आते थे, वहां पर जो सिग्नल था दैट वाज नॉट क्लियर. सो एज अ रिजल्ट वो डिफरेंशिएट करना मुश्किल हो जाता था whether there is 1T, 2T, 4T, 3T after a particular level. तो सिग्नल क्या हो जाता था? यूजलेस हो जाता था. So it was not clear, unclear. तो homopolymers के केस में virtual terminators they play a very important role because they detect one nucleotide at a time. So even if there are many T's, many C's, many A's, many G's, they will be able to detect all of them. So ये बहुत बड़ी advantage थी Helicos sequencing platform की. So the Helicos sequencing platform, like that of other sequencing single molecule sequencing platform, it involves two steps. One is the library preparation and another is the sequencing. So the library preparation step, as you can see there, the library preparation step में like that of others, you have a genomic DNA. Genomic DNA को छोटे-छोटे pieces में we get fragmented. Again, fragmentation can be done by different methods. You can use covariance, you can use sonicator, you can use mechanical shading, whatever is the case. Once you get smaller fragment, कितने smaller fragment करनी है that depends on the protocol that you are using and then what is your research question. Then once the DNA fragments are formed, they are denatured. Why they are denatured? They are denatured and they are converted into a single stranded molecule. To the single stranded molecule, with the help of an enzyme called as terminal transferase, deoxy ATPs are added at the 3' end to form the polyadenylated tail. Once this is formed, completed, then with the help of again terminal transferase, a dideoxy adenine trinucleotide phosphate is added at the 3' end to stop the polyadenylation process. This also has another advantage and we will be talking about it shortly. Okay, in case of RNA sequencing, it is almost similar to that of DNA. The only difference is that if your RNA is the starting material and if it is a high molecular weight, you can break down into smaller pieces or fragment the RNA. If your RNA is polyadenylated, this is fine. If it is not, then you can add polyadenylation tail to it. Okay, so let us assume that the RNA has poly A tail. By using the reverse transcriptase enzyme and poly-U primers, which binds complementary to the poly A tails, a cDNA is synthesized. This single strand of the cDNA is then used further for the library preparation. So this cDNA molecules again to this 3A polyadenylated tail is added using the terminal transferase and deoxy-ATP molecules, and then with the help of terminal transferase again a dideoxy-ATP molecules is added at the extreme end. So these are the last steps are almost similar. Now coming to, so this again slide, this is giving an overview of the how the fragmentation is done, that is the different steps of library preparations are done. Now what is important is जो मैंने आपको बताया है कि एक आपके पास जो बहुत ज़्यादा important है कि that you have got this dideoxynucleotide which is added at the 3' end of the polyadenylated tail. ऐसा क्यों किया जाता है? Remember this dideoxy is biopin labeled. ये आपको याद रखना है. This you need to remember. Okay, so you need to remember that the dideoxy ATP molecule is biotin labeled. So biotin का क्या? इसका what is the significance of this? That this biotin labeling, it helps in the purification or enrichment of the DNA library after the library preparation. Okay, so that is why it is very important. Moving. Okay, so once you have gotten the library, जब आपकी library बन जाती है, then what is the next step? The next step is that you would want the library to size select the library and then to purify it. Okay, so purification करने के लिए what you do is you use the magnetic beads which are called as AMPure beads. तो सबसे पहले library बन गई, उसको purify करेंगे. तो AMPure beads are magnetic beads and on these beads मैंने पहले भी बताया था iron torrent या I don't remember exactly, but इसमें पहले भी बताया था कि magnetic beads they have got streptavidin on their surface. To the streptavidin what attaches, ये जो dideoxy ATP molecule है which is containing the biotin, it gets attracted to the streptavidin molecules, and only those molecules which are having streptavidin or dideoxy ATP having the biotin, they will be purified with the help of magnetic bead, and those fragments which do not have, जिसमें लगा ही नहीं, जिसकी polyadenylation नहीं हो पाई, जिसमें dideoxy ATP नहीं लगा, they will be left behind. So this is how we enrich the library. Once the enrichment is done, the next step is to effectively take the desired fragment. So to 200 से 300 base pair के fragment लिए जाते हैं. How? They are taken up by running a gel and then cutting or taking out the sample from the gel and purifying them. Okay, so you have got two things. You have got your library enriched and then you have also taken the desired size of the library. After that, the next process, so this is how the library preparation takes place. The second step is the sample loading. So with the help of this multi-channel pipette, as it is clear here, so with the help of this multi-channel pipette, the library is loaded onto the flow cell. Each flow cell has 25 channels, and in each channel, either you can have different samples, you can have same sample also, depending upon what your research question is. And generally, 8 microliter of sample is sufficient for each well, but we put a little bit on a higher side so that as to ensure that sufficient library has been loaded to the flow cell. Okay, so once the library has been loaded to the flow cell, what happens then? अब होता क्या है इसमें बेटा कि when the library goes inside the flow cell, this is the flow cell. On the flow cell, surface of the flow cells, there are certain oligonucleotides which are already embedded on the flow cells. ये आपने Illumina में भी किया था. These oligos are special oligos containing the poly T overhangs. So these poly T oligos present on the surface of the flow cells, they are capable of binding to the poly A tails of the library, and this is how the library gets locked onto the surface of the flow cell. अभी lock हो गई यहाँ पर. Okay, so this is called as locking. So this is basically your poly T ends, and the poly T ends can bind to this blue part, and this blue part is the poly A tails. So another important feature is that अब देखिए ये वाला जो end है यहाँ पर on this side, the 3' end, जहाँ पे मैं dot